a proximal convoluted tubule, one of the important lesson with respect to the CAIE examination. Here on the right top, you can see is a photograph of nephron, and in this nephron, after this cup-shaped structure, the first convoluted tubule is the proximal. This one is the proximal convoluted tubule here. So most of the reabsorption occur here because much of the concentration of the blood is disturbed. So much of the nutrients need to be reabsorbed. So you can, as a whole, 70 to 90% of the things are reabsorbed, in which the 80% of water and 100% of glucose and 100% of amino acids is reabsorbed. It depends on the renal threshold value, that how much glucose concentration is present in your blood. If your intake is more, then definitely there will be little sugar in the urine. But if your intake is regular and normal, then there will be no glucose in the urine. Other than the diabetes patient, because the diabetes patients always have a higher concentration of blood sugar level. So the same glucose is present in the urine as well. So here we are starting that why it is the most important lesson, and for the exam as well. So you can look at this. The first one is that the lining of the proximal convoluted tubule. The lining of the proximal convoluted tubule consists of these cells. These are cuboid shaped cells here. These are the cuboid cells, right? Cuboid shape, and it has 7 different adaptations. Number 1 is the sodium-potassium pump. Sodium-potassium pump is here. You can look at this, how the sodium-potassium pump is working. ATP is required for this, and there will be a lot of mitochondria over here in the cuboidal cells. These cuboidal cells are also known as columnar cells or brush border cells, because they have number of villi facing towards the lumen of the PCT. Here there are number of villi. It is not drawn here, but there is a number of villi over here. So these villi increase the surface area. These villi increase the surface area for greater absorption of water and minerals, and definitely the glucose. Okay, so this is the brush border cells. Brush border cells are here. I have drawn these villi over the surface. Then again, now you come back to this sodium-potassium pump here. 3 sodiums are pumping out into the tissue fluid and into the blood. 2 potassiums are pumping inside the epithelial cells. ATP energy is required, that is provided by the mitochondria present here. So these mitochondria provide ATP, and there at the result of this, there is less concentration of sodium ions here in the cytoplasm, less concentration of sodium ions. This is the active transport. This will result in facilitated difference here from a co-transport protein. So this one is a co-transport protein. Why we say it is co-transport? Because it is for sodium diffusion and as well as glucose and amino acids and other organic compounds or nutrients like vitamins. So glucose and amino acids are also reabsorbed from the filtrate or from the fluid in the PCT when sodium ion diffuse inside. This is facilitated diffusion, and it is also referred to as secondary active transport. Here you can see secondary active transport. Why we are saying it is a secondary active transport? Because it is the effect of the primary active transport at the sodium-potassium pump. So this is the dotted line here, dotted line. This one is the dotted line. So the dotted line means that it is a consequence of the sodium-potassium pump, which is primary active transport here. This one is the prime reactor transport. So that's why we are saying it is second reactor transport, but it is actually a facilitated diffusion because there is no ATP required for this diffusion of sodium ions from the filtrate into the epithelial cells. Sodium ion diffuse because there is a less concentration of sodium ions here due to the active transport of 3 sodium out into the blood. So these are the two major things happening in the PCD cuboidal cells, which are the lining epithelial cells. Other than the microvilli increase the surface area. So altogether, three things. Number one, large surface area due to the cross-border cells having the villi over the surface facing towards the lumen of the PCD. Second is the sodium-potassium pump. 3 sodium pumps out, 2 potassium pumps in at the expense of ATP energy. Number 3, co-transport protein. That is diffusion of sodium ions into the epithelial cells from the filtrate, and then along with this glucose, 100% glucose and 100% amino acids are reabsorbed. Not a single molecule is present in the filtrate or later in the loop of Henry. So it depends on the, again I say that it depends on the renal threshold that there is a limitation of glucose value. If there is a higher glucose level in the blood due to a sugary meal or due to a diabetes patient case, right? So that is a limitation of the absorption of glucose. That depends on the concentration of the glucose present in the filtrate. Number 4 thing is the ATP energy that is being provided by these mitochondria present in this cuboidal cells of the PCT. Number 5 is the reabsorption of water. You can see here the water is being reabsorbed through the water channels and through the gap between the two cuboidal cells. So water is being reabsorbed from here along with this water dissolved ions and urea. Yes, urea is also reabsorbed. It is said that 50% of the urea is also reabsorbed along with the water and other minerals like calcium, potassium, etc. So 50% of urea will be excreted out in the urine, and 50% of the urea can be reabsorbed into the blood. So again and again, the process repeat, and from the whole day, urea is continuously releasing out or excreting out from the urine in the form of urine. So these are three things I'm telling you. One is that there is a water reabsorption through the water channel and through the gap. Gap is in between the two cells, and these cells are held together by tight junctions as well, although there is a gap in between these two cells. So this is the tight junction. So tight junctions, gap in the cuboidal cells, and then the reabsorption of water and reabsorption of the minerals and the reabsorption of some waste also. So altogether, you can look at there are many things I'm telling you. Again, starting from the sodium-potassium pump that is required to produce a low concentration in the epithelial cells. ATP energy is required, that is provided by the mitochondria. Then there is a sodium channels which are diffused into the epithelial cells by facilitated diffusion, along with glucose and amino acids are also reabsorbed, so it is a co-transport. Then there is a water reabsorption. Maximum 90% of water can be reabsorbed here in the BCT. And then there is a gap in between these two cells of the cuboidals of the BCT cells, epithelial lining. Then through these gaps, tight junctions are present where these cells held together, or it prevents dislocation of these cells. And then ions and urea is also reabsorbed from here. So this is all about. You can again look at these key points. Transport of protein is showing here. So this is the transport of protein through sodium potassium pump. Then ions can be diffused. So ions can be diffused from here. You can see potassium ions can because it it is becoming higher concentration in the pivotal cells. So these potassium ions are also diffusing back into the blood. And then aquaporins are there. Aquaporins for the water reabsorption. This is present in the PCT lining, pivotal cells lining, and then passive transport diffusion. Passive transport is lipid soluble substances can be done, and then primary and secondary active transport. Usually we do not name it as secondary active transport, but the facilitated diffusion, because though no energy is required for the process. So this is all about the reabsorption in the PCT. You have to write in points. Number one, at the basolateral membrane, the sodium ions is pumped into the interstitial fluid or the tissue fluid, you can say. And ATP is hydrolysis. The ATP molecules provide energy for transport of sodium ions, and this will produce a concentration gradient. And number two, the sodium ions are diffusing from the filtrate into the epithelial cells. And number three, reabsorption of organic nutrients and certain ions by co-transport by the apical membrane. So this is the organic molecules and important nutrients which should not be wasted into the urine. Then number four, reabsorption of water by osmosis through aquaporins, and the water is also reabsorbed through the gap. Also, I have told you, these solutes can then be reabsorbed as they move down the concentration gradient. Lipid soluble molecules can also be reabsorbed here through a channel protein or through phospholipids, and various ions like chloride and calcium and potassium and urea can diffuse to the gap between the two cells, which is paracellular route, you can say. So hence, 70 to 90% of the filtrate is being reabsorbed, and only water, minerals are present in the filtrate now. No nutrients is there other than this. Thank you for today.
That what is in the